this enzyme to make the more that the cell needs. And so cells need to be able to speed up or slow down enzymes to regulate them. And the terms for that are activation or inhibiting. You activate an enzyme if you speed up the rate that it's carrying out its chemical reaction, and you inhibit it if you are slowing down the rate that it's carrying out its chemical reaction. So I want to discuss some methods that the cells use to activate or inhibit their enzymes. One thing that can change the activity of an enzyme is something called competitive inhibitors. This is supposed to be an enzyme, and this little crevice right here is its active site. Here's the substrate molecule of the enzyme, and for illustration purposes, we'll say that this enzyme's reaction is to split the substrate in two, like you see there. Split like that. All right, so the enzyme's doing its thing like it's supposed to, carrying out its reaction. A competitive inhibitor is this. A competitive inhibitor is a molecule that's shaped similarly to the enzyme substrate, but is not the enzyme substrate. But it's similar enough in shape that it can plug into the enzyme's active site. Click like that. And now, since the active site is taken up by that competitive inhibitor molecule, the real substrate molecule just can't get into that particular enzyme. So the rate, the speed at which the enzyme is able to produce product molecule is of course going to be slowed because the substrate just can't fit in there. And so that's the essence of a competitive inhibitor. Molecules that slow an enzyme down by blocking its active site, by fitting into its active site so the substrate can't bind. And so all competitive inhibitors have to be similar in shape to the substrate molecule so they can fit into the active site. Now, I started to introduce this topic by saying how do cells regulate their own enzymes? And to be honest, cells usually don't regulate their own enzymes by competitive inhibitors. A cell would not manufacture competitive inhibitors to slow down one of its own enzymes. Usually, when you find a case of competitive inhibition, it's from a molecule that came from outside the organism, from outside the cell, that somehow got inside the organism and started inhibiting one of its enzymes. And to just give you an example of that, I remember reading that certain types of snake venom harm us because the snake venom contains competitive inhibitors of the enzymes that are used by our nervous system. And so by slowing down the enzymes in our nervous system, the snake venom can harm you. Here's another example for people who are undergoing chemotherapy because they have cancer. Well, the problem with cancer is some of the cells in the person's own body are growing too quickly. And so there's a chemotherapy treatment drug I should say, called methotrexate. And methotrexate works because it's a competitive inhibitor of some enzymes that are involved in cellular reproduction. So by giving the person this methotrexate, you slow down the enzymes involved in cellular reproduction, and that helps fight the cancer. So usually these come from outside the organism. Cells don't regulate their own enzymes with competitive inhibitors. So how do cells regulate their own enzymes? Well, one way they can do it is a class of inhibitors called allosteric inhibitors. And steric means shape, and allo means different. And so these are inhibitors that slow down the enzyme, but they are not the same shape. They are a different shape from the enzyme's substrate. And so what this is telling us that the allosteric inhibitors do not click into the enzyme's active site. Let's watch some allosteric inhibitors in action. Here's another enzyme. That groove right here is supposed to be the active site. Here comes the normal substrate for the enzyme. And again, we'll say that this enzyme's chemical reaction is to split the substrate in two. So, well and good, the enzyme's carrying out its normal function, but let's say the cell says, gee, we're making too much of these product molecules right here. We need to inhibit this enzyme a bit. Well, an allosteric inhibitor is a molecule that plugs into the enzyme, but not in the active site. Typically some other site in the enzyme. But when the allosteric inhibitor binds, it typically has hydrogen bond interactions or ionic bond interactions with some of the amino acids over here. And so by bonding with those amino acids, by essentially pulling on them electrostatically, it causes them to shift position slightly, which changes the folding of the enzyme slightly in such a way that the active site closes. And so now the enzyme is not going to be performing its chemical reaction because its active site has closed up, like you see there. Alrighty. So this is a more typical way that cells will inhibit their own enzymes, is with allosteric inhibitors. There's a special case of allosteric inhibitors that relates to those metabolic pathways that we just got through talking about. And it's called feedback inhibition. So let's take a peek at that. When I showed you the metabolic pathway a while ago, I used this as an example. Remember, first enzyme, second enzyme, third enzyme. And we said that this was the substrate molecule and that each enzyme made a small change substrate molecule until you finally reach the end product of the metabolic pathway like this. Excellent. All right, so let's say, just for the sake of example, that this metabolic pathway was working too well. It was making too much of the product molecules. And so the product molecules are starting to stack up like a factory that's making so many cars that it's making them faster than the cars they're selling. And so they're piling up in the car lot. That's not efficient. We're wasting resources in terms of the molecules going in, and this is certainly not doing any good to the cell to have all these product molecules stacking up. So the cell wants to slow down the metabolic pathway. Well, oftentimes what you find in metabolic pathways is this, that the product molecules are allosteric inhibitors of the very first enzyme. And so if the product molecules start to stack up, if the concentration of the product molecules gets high, eventually one of them will diffuse over and plug into the back of enzyme number one, and being an allosteric inhibitor, that will pull on enzyme number one in such a way that its active site will close. And so notice what this means now. The substrate molecule will no longer be able to enter the metabolic pathway because it can't get into enzyme number one. And so the whole metabolic pathway shuts down. It's like if you have an assembly line at a factory and the first guy doesn't show up at work, well, nobody else is going to be able to do anything that day because they all depend on the first guy doing his job. And so the whole metabolic pathway shuts down. Eventually, the cell is going to start to need these product molecules. At some point, it will. And so when that time point is reached and the cell starts using up the product molecules, eventually the cell is going to use this product molecule since it's floating around inside the cell. And notice what happens. As soon as the cell uses that particular product molecule, whoosh, the allosteric inhibition is relieved, the enzyme opens up again, and the pathway starts up. And I don't know, if you're not impressed, you should be, because it's so beautifully elegant. It's such a wonderful and yet simple solution to regulating an entire metabolic pathway. It kind of blows my mind that if the products pile up, the pathway automatically shuts down, and once the products start being used up, the pathway automatically starts up again, all because the end product is an allosteric inhibitor of the first enzyme. Need to hear more oohs and ahs out of you guys. Wow, that's really All right, so allosteric inhibitors shut down the active site, but not by plugging into the active site. There's a class of regulatory molecules that are essentially the opposites of allosteric inhibitors, and they are called allosteric activators. Here's an enzyme, and this little nook right here is supposed to be its active site, but its active site is normally in the closed position. Let's see. Okay, there's what's supposed to be the substrate, but the active site is closed, so the substrate doesn't fit in. Now, you might say, well, why might that be? Well, for this particular enzyme, its normal natural folded shape is such that the active site is closed. That's just the way this enzyme is supposed to be. That's the way this enzyme is created. For that type of enzyme, when the cell wants to speed it up, wants to activate it, there will be an allosteric activator molecule that will plug into the back part of the enzyme, not the active site. And by pulling and tugging on the amino acids, it will cause the active site to open up. And so now we have activated the enzyme, and it will start carrying out its chemical reaction. As long as the allosteric activator is there, the enzyme will be active. All right, and there's a related type of activation that you see in other enzymes called phosphorylation. Here's an enzyme, and this is another example of an enzyme whose natural shape is such that the active site is closed, not open enough to grab the substrate molecule. For some of these enzymes, what can happen is the cell can add a phosphate group to one or a few of its amino acids. And remember, phosphate groups are negative ions, and by changing the R group interaction, because now this one's going to have a phosphate on its R group, by changing the interactions of the R groups, the tertiary structure, the folding of the enzyme changes, and that opens up its active site, and now it's going to start being able to carry out its